Hello students, this is Jayashree and in this video we are going to discuss about cell wall. We already know that animal cells lack this cell wall. So, it is the one which forms rigid outermost covering of plant cells, fungi and bacteria. And in case of plant cells, it is made up of cellulose. Now, what is this cellulose? Cellulose is a complex substance and it provides structural strength to plant cells. Now, what about this fungi and bacteria? In case of fungi, the cell wall is made up of chitin. In case of bacteria, it is made up of peptidoglycan. So, the chemical composition of cell wall in these three organisms are different. And we already know that cell wall is located outside the plasma membrane. Now, moving on to the functions of cell wall. Just like the walls of our house provides us protection, likewise, even cell wall provides protection to plasma membrane as well as the other inner components of the cell. Other than this, it also provides definite shape and structure to the cells. Now, if you observe, these two functions are common for plasma membrane as well as the cell wall. Moving ahead, we have one more function. For that, let us see what is going to happen when we take an animal cell and place it in hypotonic solution. So here I have drawn a circular animal cell and I have shaded it in red color and we are placing it in hypotonic solution. Then endosmosis is going to take place, that is the water from the external media is going to enter inside the cell and as a result the cell is going to swell up. Now next what is going to happen is that the water which has got accumulated inside the animal cell, I should say actually the extra water which has entered now, right? That water is going to exert pressure on the plasma membrane. Remember here there is no cell wall, right? So that water is exerting pressure on the plasma membrane and beyond a limit plasma membrane cannot stretch, it cannot withstand that outward pressure and it ruptures, releasing the cellular content resulting in the cell lysis. So on the whole I can say if I take animal cell and place it in hypotonic solution, finally cell lysis is going to take place. Now let us see what is going to happen if I take a plant cell and place it in hypotonic solution. So here we have a plant cell and it has a cell wall. We have placed it inside the hypotonic solution. Again the process of endosmosis is taking place, see, and the cell is going to swell up and this time the cell lysis will not take place, rather the cell is going to remain turgid. Now what is this turgid? In biology, turgid term refers to cells or tissues that become swollen due to water uptake. So we say plant cells become turgid when we place them in hypotonic solution. Now the question arises, why there is no cell lysis over here? The answer is very simple. The extra water which has entered inside the plant cell, it exerts pressure in the outward direction on the cell wall and in return the cell wall also exerts pressure in the inward direction. So these two forces, they cancel out each other's effect and as a result more or less the cell is going to remain stable. Now we need to understand about a process called as plasmolysis. Oh, what is this plasmolysis? Now this process is seen exclusively in case of plant cell. So this is a normal plant cell. So it has got a cell wall which I have shaded in black color. Then I have drawn plasma membrane in blue color and this light blue color region which I have shaded is cytoplasm. This cytoplasm contains a nucleus and certain cell organelles. So these four cell organelles I have drawn, and this huge structure what you see basically is vacuole. Vacuole is also a cell organelle, and basically one of the important function of vacuole is to store water. So it contains water plus other solutes, and sometimes it may occupy about 60 to 80% of the cell volume. Now if I take this plant cell and place it in hypertonic solution. So here we have placed plant cell in hypotonic and we saw what happened. Now we are placing plant cell in hypertonic solution, which means external media is going to be concentrated. As a result, exosmosis will take place, which means the cell is going to lose water, and mostly it is the water which was stored in vacuole which is going to come out, and as a result you can see the entire cellular content of the cell is going to shrink and move away from the cell wall, and this process is called as plasmolysis, and the cell which has undergone plasmolysis is called as plasmolysed cell. Now, one interesting thing is that this process of plasmolysis is reversible. Now, how it is reversible? If I take this plasmolyzed cell and place it in hypotonic solution, again the process of endosmosis will take place, and this cell is going to regain the lost water, and all the cellular content which had folded will revert back to their original position, and this process is called as deplasmolysis. That's all for this video. Thank you.
Do like, subscribe and share this video.